Hi everyone, I'm Christian Chevalier. I'm a research director in the Food Biology and Pathology Laboratory at INARE Bordeaux, southwestern France. Bordeaux region is well known for wine and its grapes, but my favorite model for research is tomato. So today my talk will be centered about a process that is involved in fruit growth in tomato that is called endoreduplication, and I will detail what is this phenomenon. So first of all, I would like to give you some definitions. What is endoreduplication? Endoreduplication is in fact the most common form of endopolyploidy in flowering plants. It occurs in 80% of angiosperm species. And by endopolyploidy, we hear, in fact, the occurrence of different ploidy levels, namely nuclear DNA content in an organism. So endoreduplication, in fact, is a modified cell cycle, and it consists of one or several rounds of DNA synthesis in the absence of mitosis. And as a consequence, this leads to the production of polytenic chromosomes that are characterized with 2N chromatids still attached together, and this production of polytenic chromosomes do not produce, in fact, a change in the chromosome number inside the cell nucleus. This phenomenon of endoreduplication, as I said, is quite common in most angiosperm species, and here on this slide you have some illustrations of some very well characterized system of endoreduplication. On the left part, you have, in fact, several organs in Arabidopsis. Endoreduplication happens in flowers, it happens in stems, it happens in roots, it happens in trichomes. And in fact, when you use a flow cytometer to determine the DNA content in the nucleus, you end up with this kind of graphs where you can see, in fact, the evolution of the DNA content, a 2C DNA content, which corresponds to a diploid state, 4C, 8C, and 16C. So this is the particular characteristics in Arabidopsis. In another system, which is very, very well known, which are the legume root nodules, where you have these nodules developing after the interaction with some bacteria, which will in fact produce these nodules that are really important for plants, as they are the nitrogen fixing organism here. And when you look at these nodules in a flow cytometer in the nucleus, and then you have all these different peaks increasing. Another system in a monocot plant is in the albumin in maize grains, where you have in this central part, again, this very important DNA content. Now, if you look at the tomato, the fleshy part of the tomato, which is called the pericarp, you end up with this kind of graphs, and you can see here the absolutely enormous amount of DNA content that can be reached at the end of the fruit development. It's almost 512 C of DNA content. So all these systems have in common this increase in DNA content inside a nucleus, and in fact you can see that it's also related with cell size. It's an enormous increase in cell size related to this enormous increase in DNA content. So if we look at the developmental process that is endoreduplication, here you have a classical cell cycle, which is related with cell division, and especially with this mitosis phase. The cell cycle is divided in 4 phases. You have the diploid state in this G1 phase, then you synthesize the DNA in the S phase, you end up with the G2 phase, and then during the mitosis, you have back a return to the 2C phase because you have the release of 2 daughter cells. In the endoreduplication cycle, you don't have anymore the mitosis phase, and then it turns like that, and it produces 4C DNA content, 8C DNA content, 16C, and further and further on. And then that's the typical graphs for an endoreduplication system. And this is related with cell growth, with cell expansion. A according to the mitotic cycle, in fact, after the metaphase, during the M phase, you will have the anaphase where the duplicated chromatids will be separated. At this phase in the cell cycle, you will have a 4C DNA content, but you will have, in fact, 4 chromosomes here. And then in the telophase, you have the separation of the 2 daughter cells, and you return back to a 2C DNA content and a 2N number of chromosomes. During the endoreduplication cycle, there is no real metaphase and there is no anaphase. You don't have the separation of the chromatids here, and you keep a 4C DNA content with a 2N number of chromosomes. And if you make another round of endoreduplication, then you will have, in fact, always 2 chromosomes here, but you will have a 8C DNA content. If we think of a system that could, in fact, detect all the chromatids, all the duplicated chromatids on the chromosomes by, for instance, hybridization of a molecule, if you can have this kind of a system, you will have this detection of the chromatids that could be, in fact, according to the chromosomes, in 2 different clouds. So we use, in fact, this kind of a technique, which is, in fact, the fluorescent in situ hybridization technique, the FISH technique, to make an hybridization on these chromosomes and see how you can, in fact, detect the different DNA content levels. So what we have done here is to take fruit pericarp tissue. We purified the nuclei, then we sorted this nuclei in a flow cytometer. We put all these nuclei on glass slides here, and then we can observe it under a microscope. You can see a 4C DNA content nucleus, 8C, 16C. You have a nice increase in the size of the nucleus according to the DNA content. And then we think of a probe that could be a long DNA sequence, let's say 100 kilobase pair, that could be specific of 1 chromosome here. And then we hybridize this probe with this purified nuclei. And we can here see that we have, in fact, the amount of dots corresponding to the different chromatids. 1 chromatid, 1 locus would be 1 dot. And in fact, according to the DNA content, we have the increase in the number of dots, and we have the separation of these dots in 2 clouds, which correspond, in fact, in 2 chromosomes. So we wanted, in fact, to use this technique to detect the ploidy levels inside the tissue, and that's what we have done by studying, in fact, the tomato fruit development. If we have a wider aspect of this tomato fruit development, this is the evolution of the size of a tomato fruit according to development, according to the age. If we make cross sections of the fruit and if we measure the cell area, we can see that according to development, there is a very important increase in the cell size from the flower to the ripened fruit here. And if we measure, in fact, the mean DNA content value in the nuclei, we have also this increase in this content of DNA. So there is a correlation between the increase in cell size and the increase in DNA content. On the right part of this image, we have done, using this FISH technique I talked about on the previous slide, we have used this technique to, in fact, detect what is the DNA content in all these individual cells across in this cross section of the tomato fruit pericarp. And we have been able to assign to each size of cells a DNA amount. Of course, it's a range of DNA amounts, 2C to 4C for the smallest ones, and for the biggest one here in the central part of the pericarp, we have the largest amount of DNA content, 128C or 256C. So the idea about here is that endoreduplication is really involved in cell growth. It's a DNA-dependent growth for cells, and consequently, having more cells of bigger size will have a consequence on the size of the fruit. And so endoreduplication has a consequence on the determination of the organ growth. Interestingly, what happens in different species of fruits? And we have plotted here the duration of fruit growth of a whole category of fruits according to the number of endocycles, of endoreduplication cycles, which correlates to the DNA amount inside the nucleus. And what we see here is that for all the species that requires a very small period of growth, they have this endoreduplication process occurring. On the contrary, fruit species that require a long duration for growth have never endoreduplication in their nucleus. So we believe that endoreduplication is a trigger for growth, is an accelerator for growth during fruit development, and is particularly true for Solanaceae species like tomatoes, potatoes, or other species. So of course, this endoreduplication process is multiplying the DNA amount in a nucleus, and it multiplies, of course, the amount of genes. So is there any relationship between endoreduplication and gene expression? If you have more genes, are there more expressed? So to answer this question, we've made several experiments. The first one, in fact, was to follow the amount of the RNA polymerase II, which is the RNA polymerase that transcribes genes into messenger RNA. And we have a clear correlation between the amount of proteins and the DNA content inside the nucleus. Secondly, we have made, in fact, not a DNA fish experiment, but an RNA fish experiment using a ribosomal RNA probe to measure, in fact, inside purified nuclei, what was the result of the transcription of this rRNA gene. And we clearly see here in this green dot that we have an increase in the transcription of this gene according to ploidy levels. If we extract the total nuclear RNA content from purified nuclei at various ploidy levels, we have also a clear increase in the total nuclear RNA content, meaning that the whole transcription of RNAs, ribosomal RNAs, tRNAs, messenger RNAs, are increased according to the ploidy level. And this is also true if you do some gene expression studies using Q reverse transcription, PCR, and some of genes, and we clearly see here an increase in the gene expression according to the ploidy level. So clearly, endoreduplication induces more activity at the transcription level. And what we wanted to demonstrate in the following experiment is to have a wider view, a genome-wide view of transcription. So here, what we did was, again, to purify nuclei at various ploidy levels, 4C, 8C, 16C, 32C. We had a fixed amount of nuclei sorted by flow cytometer, 100,000 nuclei for each of these ploidy levels. And we did an RNA sequencing experiment starting from this amount of nuclei. We are not basing our RNA sequencing analysis on a fixed amount of RNA, but we are basing our analysis on a fixed amount of nuclei. And through this experiment, we have been able to demonstrate that we could have more than 1,000 genes that could be differentially expressed when we compare, in fact, a 4C DNA level to an 8C DNA level, an 8C to a 16C, a 16C to 32C, and so on. And we have been able to follow the expression of these genes according to the different ploidy levels. And we have all these modifications here. We have genes that are that are preferentially expressed at low ploidy levels. We have genes that are preferentially expressed at high ploidy level. And according to the ontology of all these genes, we can assess some biochemical functions to these studies. And then what is extremely interesting here is that the activity of the cells in terms of gene transcription is in fact related to their ploidy level, but also to their location across the tissue. And you have the smallest cells here that have a particular metabolism, and then you have a highly developed cells here that have another metabolism. So these experiments were very interesting in demonstrating that underreduplication has a real impact on transcription activity and a real impact on the differentiation of cells according to development. So as a take-home message, we can say that in fact the role of underreduplication during tomato development is in fact a mean to accelerate fruit growth. It's also a mean to control cell size, and especially according to the theory, which is said that the nucleus to 5%, the chiraplasmic volume ratio theory. And it's also a mean to promote enhanced gene expression. And this is in connection to sustained metabolism. You have more genes for more enzymatic activities, and finally for more metabolites, and in a cell differentiation status. So I thank you very much for your attention. And if you have questions about this presentation, then you have my email there, and I'll be happy to be in contact with you. Thanks very much.